This research summary is on a VTA to basal amygdala dopamine projection, contributes to signal salient somatosensory events during fear learning, and it's going to be on figure 3. The study is performed by Wai Tang, Alexei Kobuchi, Michael Kincher, and Ralph Schnegenberger, and is annotated by Aarman, Madison, Peter, and Brooke. So some background information for this research is that experiments regarding fear learning and condition response have long been studied in the biomedical community. Some key research, including Ivan Pavlov's famous experiments, who first introduced classic conditioning, and M.R. Papini. These experiments have most often focused on the neuroplasticity of the brain and corresponding areas of the brain that induce those fear responses as a result of conditioning. The goal of this research is to study the role of dopamine and dopamine transferring neurons in fear learning with the VTA in the basal amygdala, with a variety of different factors, including optogenics and inhibitors, which will be expanded on later in the presentation. This research is very significant to the biology community, generically for a better understanding of the role of the VTA in basal amygdala and their connections to other mechanisms of the brain and involuntary neural responses. The study also has more applicable significance to the biomedical community. It can be used in fear conditioning and the biomedical aid of treating fear condition disorders like PTSD and ordered anxiety. Additionally, this research also builds more background information and knowledge and study for potentially groundbreaking future research addressing fear condition responses, which can result in pseudodementia or memory impairment stemming from a fear-induced trauma. Some of the major biomedical questions that this research addresses are: what is the role of dopamine projection in the VTA in the basal amygdala? Since it is well known that these areas of the brain have dopamine releasing neurons, the ultimate goal is to determine how do these parts of the brain play a role in aversive fear learning. Since there is less study on the VTA and the BA in fear conditioning, even though it is considered to be a major area of the brain in developing these responses. Are the VTA and BA connected in fear learning? The goal of this question is just to touch on the potential connection between the two parts, since there's more focus on what they do individually and less study on how they function together in fear learning. How does dopamine release play a role in fear learning? And for future application to the biomedical field, a good question for potential further research could be, could dopamine treatments potentially be employed to benefit trauma victims and others with negative fear learned responses?
Before we get into figure 3, let's talk about figure 2 first to give a little bit of recap. So basically, there were dopamine producing neurons in the VTA that was acquired in response to the foot shocks. The data basically displayed the higher responsiveness to unconditioned stimulus in the VTA, basically displayed that the neurons of the mice and the resilient AP were formed. The firing rates after training the CS in the US proved that the CS was successfully trained to the US in majority of the mice, and that the CS did the fear response by day 3, even without a foot shock actually being delivered. Comparing it to day 1 and day 2. In day 1, there was a tone, but no foot shock in the AP and no fear response. In day 2, there was a tone, a shock, and a fear response in the AP. To wrap all this up, this all resulted in the VAB dopamine neurons play a significant role in the auditory fear of learning. So in research, the research questions for figure 3 are 3 main ones. Do BA projectors increase the activity of tone shock pairing with CS and US in comparison of the control group just only being the CS? Are the basal amyloid gland projectors dopamine neurons? And are the BA projectors among D18 neurons respond to the foot shocks? So the overall workflow for experiment figure 3 is that the CTB is ejected into the mice. Once that's ejected, there'll be a 3-week recovery for the mice. Then after that, there'll be a 1-day inhibition where we apply tone blocks into the mice. Once a day passes by, there'll be 2 days of training to see if the tone blocks are being expressed, and following that with foot shocks. After the 2-day training, mice will get killed and will be prepared for the Sophis immunohistochemistry. Our control for experiment 3 is the mice only having the CS, or the conditioned stimulus. This will help compare and contrast the differences between the CS and the CS-US combination that we project that the BA projectors will have an increase.
So the results of figure 3 in A basically shows a preparatory stage for the experiment where the mice receive a CTB injection, and then there's a 3-week rest period, and then they undergo a 90-minute training period, then are killed and prepped for the CFOS immunohistochemistry experiment. In B and C, these images display a control mouse and a CS-US pairing mouse. Both of these mice receive the injection, and which is seen on the upper left side. And mouse in B only receives tones, while in C, the mouse receives tones and foot shocks. The right box right here displays the DAT positive neurons in the VTA, which are identified by the TD tomato signal. The bottom images show individual concentration on the channels, and then on the far right, they're merged together. This identifies the fluorescence image of the BA projectors, which is the cyan channel based on their CTB fluorescence injection, which is seen as the magenta color. And this helps review the co-location it has with the CFOS antibody, which is the yellow channel right here. And D and E gives the results of the VA projector numbers that are 73 and 63. In D, the control mice had 73 VA projectors in total, and only 2 of those were non-DAT cells. And one of those 2 cells did not result in a positive CFOS antibody, and the other cell was positive for CFOS, which corresponds to the BA projector at 1.4%. Of these 73 projectors in the control group, only 11% of the CFOS was positive, which is seen in this portion right here. And in E, the CS-US pairing group fraction was greatly increased at 43% of mice positive for CFOS. The number of non-DAT projectors compares to the control group, as it is still on the small side at 3%. The results of the CFOS label experiment shows that the BA projectors do activate quite noticeably during the CS-US pairing. For the TD tomato fluorescence of the DAT positive cells support that more than 95% of BA projectors are in fact dopamine neurons. The result of this experiment provides evidence that supports the vivo tried recording experiment and suggests that the basal amygdala projectors are amid the units providing an increase of the action potential firing in response to the foot shocks.
All right, these are the conclusions that are being drawn off of figure 3. If we look, the projections from the BA to the VTA are the same across the experimental mice. So we're looking at these magenta spots here. We've got 4 to 5 in the top and 1 to 2 in the bottom. So there's basically the same biological connections in these mice. However, we do see the BA projectors showing an increased firing in response to the unconditioned stimulus. So the VTA is sending more signals to the BTA, to the basal amygdala during the unconditioned stimulus. So we see much more CFOS when the conditioned and unconditioned stimulus are paired than in just the conditioned stimulus, the sound. When you add the foot shocks, there's a lot more activity, not in the DAT as well as the CFOS. So with about the same physiological connections. So definitely there's a lot more brain activity going on in the basal amygdala based on the connections to the BTA. These dopamine sending connectors are doing a lot more when you've got this connection going, which suggests that the VTA projections to the BA, the basal amygdala, are involved in learning of fear. These connections are popping off when the stimuli are being paired, which is when the mouse is learning to make this fear connection. When they hear this noise, there's going to be foot shocks. I need to be scared. Criticisms of figure 3. It's very opaque. The figure relies a lot on abbreviations and acronyms. So you're going to struggle potentially viewing all this information, which is great information, but viewing it without knowing necessarily what the CS and US pairing means, what the CFOS or DAT is, what that comes from, you're going to be left very confused. So you need to be reading the supplemental information in order to understand what any of this means. Understand that the significant increase in the CFOS and the DAT in the paired mice means that they're learning, that the connections, the VTA BA connections, mean that they're learning and that they're involved in learning. So like what would really help this a lot is a key describing very explicitly what the cyan, the magenta, and the yellow spots in this image are meaning. It's there, but a little key in addition would significantly improve the readability of these figures. And then finally, we've got our references, and that is figure 3. Thank you so much.